anyway. So we met last night, most of us. I'm Heike, I'm a biochemist. I used to work in New York and I recently moved back to Europe and I'm working in Italy right now. I have been studying CK2 for 13 years and I said that last night already. I've never spoke to a person ever that was really interested in CK2. So now being here and being amongst you who have an interest, who have a true and important interest in the kinase makes me feel really wonderful. I'm so glad that I can be here, and you being here makes me also want to work much, much harder than I have. So I will keep working on CK2 forever. I called my talk CK2 Jack of all trades because Volkan has already told you there's a huge literature on the function of CK2. And when you Google Okur-chung syndrome, you won't find much, but if you Google CK2, you'll be overwhelmed. And you'll learn quickly that CK2 is a fundamental protein expressed in every cell of our body that is doing so many things that we could talk weeks about it. So that's why I called the talk Jack of all trades. And I'll start quickly talking about the history of CK2 research, since it's a long history. I'll talk about the protein that is CK2. I'll talk about the protein's function in our bodies, in health and disease. And then I'll talk about what it could mean to have CK2 reduction of activity in our bodies, which is most probably what we're talking about with OCNDS. And I'll tell you about my plan, what I'm going to do and start doing as of September on my work done on OCNDS. So here you see that work on CK2 started in the late 1900s. In 1883, a Swedish chemist actually found that a protein, a milk protein called casein, contains phosphorus. And that was a new thing for biochemists. A protein should be a protein. It shouldn't be a phosphorylated protein. It should not have phosphorus. And so since then, the search was on to find out what actually is happening in the body or in cells so that phosphorus gets onto that casein protein. And in the 50s, a British researcher, Burnett, found out that when you take liver extracts from a human liver or cow liver or whatever, and you put it together with casein, and once of a sudden that casein gets that phosphorus group again. So the activity of putting phosphorus onto casein was found in 1950. And ever since, in the next decades, we understood more who that thing is that does the job. It's called CK2. It's a protein that is of a specific function. The function was identified, the genes were cloned. CK2 was found to be highly expressed in the brain, for example. We understood a role in cancer, and this is our point zero when we're all starting to, gear up. CK2 is actually linked to a human neurodevelopmental disorder. So in 2016, I think it was really an amazing paper, and we all start from there, from Okur-chung paper. So Emily has already told that the gene encodes for a protein, so I can go through that very quickly. Basically, I'm starting here where the protein is being made. DNA encodes for amino acids that are put together in a chain, in a long chain. But since these amino acids are molecules that have electrostatic properties, the molecule will not be just a chain, a long one-dimensional chain, but these electrostatic properties, they mean attraction, repulsion, so that one chain becomes a globule, becomes folded, and it becomes a three-dimensional structure. And then it becomes a functionality, it becomes something that can do a job. It couldn't do it as a single chain doing nothing. It has to have a structure. Sorry, I'll try that again. Is that not good? No? Okay. So, just as a recap, CK2 is a protein. We know that by now. CK2 is more than that. It is an enzyme. An enzyme is a protein with an activity, with a job to do in the cell. And the activity that CK2 does is to be a kinase. And that means it puts a phosphate group onto a substrate protein. A substrate is the receiving end of the job. So the phosphorus is taken up by CK2 and put onto a substrate protein. And for CK2, you'll see it is a large set of substrates that CK2 has. Why is phosphorylation important? What is special about it? Well, it's the most fundamental mode of modulating an activity of an enzyme that's quickly reversible. So basically every cell needs to respond to challenges, stimuli. If you see something, your brain reacts very quickly, you run away. So you need a mode to regulate proteins that are already made in the cell to make them active and inactive very quickly. And phosphorylation was one of the first modes of regulation that showed us that we can actually, our bodies can manipulate activities very, very quickly by adding a phosphate group onto a protein, this globule thing, a protein, thereby changing its structure. And by changing the structure, again, through electrostatic attraction and repulsion, the protein will behave differently in the three-dimensional environment that a cell is. It will encounter its partners in a different way when it's folded like open or like closed. So this is important, and the phosphorylation changes the structure of a protein. And then it can be, if the cell decides that that's enough of the signal being propagated, we want to stop it, phosphatase, another enzyme, comes in to undo the job to dephosphorylate the substrate. And this is so important. This mechanism is underlying many, basically all functions, all physiological processes in our bodies, from metabolism to thinking to running to sleeping to whatever you can think of, to sex. And it was awarded in 1992 at the Nobel Prize because it is so fundamental. Here is just a little scheme that shows you that CK2 is not the only kinase in our body. We have 518 altogether. Almost 2% of human genes represent kinases, so it's an important mechanism. And CK2, amongst all of these, well, it's only the green little family here, just alpha-1 and alpha-2, CK2 alpha-1, CK2 alpha-2, but in contrast to many other kinases, CK2 has a vast array of substrates. And that is, trying to link it back to OCNDS, also something that makes it, maybe explain the variety of symptoms, because CK2 doesn't only work by phosphorylating one specific pathway or enzyme or protein, but it has hundreds of them. Therefore, it's sometimes being called a housekeeping gene, which is not quite right because it's still part of, regulation, etc. But it is so present and so important. 300 or more proteins in the cell are being phosphorylated by CK2. And by the way, CK2, historically called like that because it is phosphorylating casein, is actually not casein kinase in real life. I hope I don't confuse you by that, but when those experiments in the 1950s were done, everything was still in the test tube and nobody was able to work yet in vivo with mice or even cell lines really. So now we find out that while CK2 can phosphorylate the casein protein very well in the test tube, in our mammary glands it's another protein, another kinase that does the job. But the name stuck to it and we just now call it CK2, full stop. We don't really say casein kinase anymore. So back to the structure. CK2 has a wonderful structure, as you will see in the next slide. Here again is the primary amino acid structure, which will fold into a two-dimensional structure, like here, and then into that globule, the three-dimensional structure. And for some proteins, that is not enough. They need to find a partner amino acid strand to form a complex, a quaternary structure. And that is the case for CK2, and that was described. Here we go. I think it's a Mac PC issue here, but it doesn't matter. You still see that CK2 is a beautiful butterfly-like structure that has 4 different colored amino acid chains. So basically 4 proteins come together to form this holoenzyme called CK2. 2 of these units here are the catalytic subunits, the alpha, casein kinase 2A1, or also A2. Here, those 2, these are the catalytic subunits. And then we have 2 beta subunits, which are what we believe to be more regulatory. For example, they guide CK2 to find their binding partners and localization or substrates. So the ones that do the actual job are the alpha subunits. And since we have 2 genes for alpha, we have alpha-1 and alpha-2, and OCNDS seem to be caused mainly by alpha-1, and there is not an alpha-2 yet described. And that makes sense in a way because CK2 alpha is the predominant one in our cells. So you have alpha prime, but it is expressed at much lower levels, maybe by a factor of 5 or 10 or something. But it could be imagined, and that I think should be studied for OCNDS, whether alpha prime can be regulated as well in its expression, alpha 2, whether that might be altered in patients of OCNDS, because it can be compensating for the action of alpha 1. But this is to be studied. So Emily gave the most beautiful talk about what mutations are and what they can cause, so I'm not going to talk about that. I'm going, for example, here she talked about a mutation that leads to a stop of the enzyme, or she talked about mutations that where simply one building block was changed and led to a different amino acid to be used. And this is just from a structural point of view, it shouldn't confuse you much, it's just an example of any generic protein. But when an amino acid that is, for example, large and bulky, like this one here, phenylalanine, that has a ring, when that is changed with by a small amino acid, you can imagine that here that fold might collapse because the space that has been taken up by this large amino acid is gone, and therefore that will affect the structure, just as phosphorylation itself will affect the structure. It's always about electrostatic properties that hold this three-dimensional structure together. So this is what mutations can cause. Even if they're just a single little point mutation, they can cause the structure to be altered. And here's the CK2 alpha. I'm sorry about that. This is the protein chain, like the one-dimensional protein chain. We have 389 amino acids in a line for the alpha 1. And you see here, I marked some regions in red, and they're simply put in red because we believe that they're very important for the function of CK2. For example, they could be important for holding the conformation, the three-dimensional structure in the right place, or they might be important for binding the substrate, or for the transfer of that phosphorus group. So one could imagine that if a point mutation happens in one of those red marked areas, that this would have a stronger effect than maybe if a domain that's gray and we don't really know very well what it means for the function of the protein, but that would entail. Probably a mutation in the gray area would mean much less in terms of physiology. So what happens what does CK2 actually do in our bodies? To understand that, we often use the knockout approach, where we delete the gene to see what then happens, what then goes wrong. And that was published as a paper by Lu et al. From Boston in 2008. They were trying to knock out the alpha-1 subunit, so basically make a mouse that doesn't have any CK2 alpha-1, 0. And it is kind of to be expected, because CK2 is highly expressed in every single cell, that mouse is not going to make it, and it dies in the first third of gestation, with mainly problems with the neural folds, with the brain, and with heart. And it's smaller overall, and at early age it's absorbed into the uterus, so it doesn't make it. So we learn from that CK2 is expressed very early on in embryonic development. It is important for brain, heart function, but probably much more. It is just like every time you search for something, you focus on something and you don't look at other things because you have to start somewhere. So, but those researchers claimed mainly heart and brain need CK2 in early development, right? And CK2 has been studied in the last 15 years with a heavy focus on cancer, because many cancers have been identified to overexpress, to have more CK2 than normal. For example, here, this is a paper from 2014. You see that this alpha-1 subunit that we're also talking about in OCNDS, in certain cancers you have much expression of CK2 alpha. Like the breast cancer has a 20-fold overall overexpression of CK2. Then we have head and neck cancer, a 10-fold, 14 for lung cancer, and in the case of the female cancers like breast and ovarian cancers, the clinical outcome is also adverse. So the more you have CK2 overexpressed, the lower the survival chances are. So because you're a patient group and I'm not a doctor, I can't make statements, but this shouldn't alarm you because it's the opposite of what we expect to happen in the OCNDS. In OCNDS, as you'll see later, we're not talking about overexpression, we're talking about most probably loss of function of CK2. So if I could extrapolate naively from my biochemical knowledge, I would say that where OCNDS patients are, if anything, maybe rather protected from these cancers because they're not going to be overexpressing CK2 to these high levels that would drive the aggressiveness of these type of cancers. So CK2 was studied in a lot of mouse models, of course, on the tissue culture in its capacity to drive cell growth and cell proliferation. And a lot of mouse models have shown that when CK2 is pharmacologically inhibited and mice are given human cancers, like you can implant them under the skin, that when CK2 is inhibited, the mice do much better, the cancers don't grow. So CK2 is a driver, and when CK2 is inhibited, is protected from driving the cancer. And there were human studies as well using CK2 inhibitors, that showed that CK2 inhibition can be beneficial for certain type of cancers. We are interested in studying CK2 in the brain. CK2 is highly expressed there, and the activity of CK2 in the brain is very strong. So here's just a slide that shows you that all of these units, alpha 1, alpha 2, and beta, are expressed in the mouse brain basically everywhere you look. It's important in the brain. And we are studying in particular, I'm not really talking about that because it would go too much into detail. We're studying CK2 as a modulator of signaling in the brain. For example, as a modulator of responses of our brain to dopamine signaling and serotonin signaling. A little bit I show you in a few minutes about the work, but not too much. So what happens if an organism really lacks CK2? I already showed you the slide where CK2 alpha 1 was completely deleted from the genome and therefore prevented from being expressed. The mice are not viable. When the alpha prime, the alpha 2 subunit or gene is knocked out, the mice are actually doing very well. They have no obvious phenotype. The male have a defect in spermatogenesis, but other than that, they're doing well. And the CK2 beta mice that don't have CK2 beta, the knockouts for beta, they are also heavily affected, and the CK2 is involved in prenatal development, and hence the mice do not make it to be born. So at the time we decided we need to make a mouse that allows us to study CK2 function in not the embryo, but in the adult or adolescent animal. And so we made an approach to knock out CK2 specifically in cells that we are interested in. So it's called a conditional knockout. And you do that by making a transgenic mouse, or a mouse that, now I hear myself better, and you breed it with another driver that targets the cells where you want CK2 to be ablated, right? So we were interested in CK2 in its role in motor behavior and also mood, and so we did a certain knockout. We knocked out CK2 in one part of the brain. The bigger the part of the brain was that we targeted, the mice were not viable. But when we chose just specific cells in the brain, the mice were viable and they grew to adulthood and we could characterize them. And you see some of this characterization later. That means, again, CK2 is super important. And from the work that I showed you before, from the CK2 alpha knockout mice, the heterozygous mice, they are fine. The heterozygous mice, as we know from Emmeline's talk, they have 50%, they have one allele that's correct, and one allele here by the knockout that's not present. So they express 50% of CK2 wild type, non-mutated, perfect CK2, and they're doing just fine. They have a one defect that is compensated, and so nobody, as of now, has studied this heterozygous line because people just said they're doing fine, these mice. Now I think, actually just in preparing the talk, I'm like, why did I not get these mice earlier and look at those mice, because they're doing fine, your kids are doing fine. If you know what I mean? If you don't say, okay, we're looking specifically at one cell type, we're looking specifically at one phenotype, at one behavior, you might not detect that these mice have something, and they might be actually a quite good model to start, understanding OCNDS that we have now on hand. We're making more, and Dr. Chung is helping with that a lot, but for now, this is the line that's available, and that's possibly coming close in symptomatology. So again, here are, that's CK2, again these 389 amino acids, and here are the mutations collected from the various papers. The first original cohort here, the 5, and then the subsequent ones. And you see that a lot of these mutations are actually occurring in the red areas that I told you before are important for maintaining function. And this is why we expect some phenotype from those mutations. But not all of them do. Some are in the gray areas, and yet we see a phenotype. That means we cannot just predict by looking at the structure what the phenotypical outcome is. We have to actually study every single mutation by itself. And so we can speculate at this point that for very probably, most probably, patients that have OCNDS have 50% of the healthy protein because they're heterozygous. Then there is the mutated allele that we have to study in detail. For some mutations we can already predict, for example, that it might not be as stable, so there might be less protein there than normally is because it's made and being degraded faster. For example, it has been shown by people in vitro, not in vivo, that N-terminal truncation or changes in the N-terminus on the left side of the protein lead to less stability of the protein. Okay. Another mutation suggests that the stability of an intermediate might be affected and therefore less protein might be made. And then most probably we'll talk about activity being changed because it will the point mutations in those active sites, in those important sites, will alter the three-dimensional structure of the active loop or whatever you want to call it, and they will most probably reduce activity. For example, the D15H mutation definitely will have abolished activity. So that patient probably has 50% of activity or protein present, and that's probably all, but we need to test it. Then mutations, just as a 198R, which there are several patients who have that should affect the binding of the substrate and would most probably reduce it, but we don't know to what extent. Maybe the extent is very mild. This we'll have to test. Then mutations in another loop to the left side are also predicted to reduce activity, etc. But there's one mutation, this is not a disease mutation, but that's a mutation that was made by a biochemist in the lab, but it's very close to one that was in the original paper. This mutation is increasing activity, so theoretically we can expect everything from decrease, most probable, but we can also possibly find one or two mutations that will increase activity, and hence, we have this huge spectrum of phenotypic traits. So what we're going to do very soon and we're ready to start, is to try to understand what each individual mutation does. First of all, to the activity, so how does it affect the CK2 doing its job that it's supposed to do, which is phosphorylating substrates. We will do that in the test tube with purified proteins for every mutation that has been described. And then we'll move on to transfer this mutated plasmid or whatever into cells, where we will take the endogenous activity of CK2 off, so we can study in the cell what the mutant actually does. We can study different substrates. We will pick the most important of these, or what we believe are the most important, for which we have the tools to study whether they are less phosphorylated or even more phosphorylated in the presence of this mutant. And of course, we'll check other things such as stability. Where does that mutant sit in the cell? Maybe it has a preferential localization that's different to the wild type, to the non-mutated version. We'll do all that with tissue culture work. But the next step, and that's where you all come in and can help us, and that's definitely the most important part for us as researchers as well, is to work with the cells that come from patients, and we'll do, similar work. We'll check for which substrates are affected by the mutation, in what direction, what does that mutant, how does it behave in the cell, and how does it affect cell behavior. For example, if we have skin cells, we can do a lot of migration tests, we can do wound healing tests, we can see how the cells look. The morphology of the cell might look different. So we'll have an open mind about addressing various things with these patient-derived cells, and then we'll have hypothesis-driven experiments. For example, for us it will be brain. We will, or Dr. Chung again has initiated that patient-derived cells will be made into what are called omnipotent cells that can be driven into differentiation to become brain-like cells, and then we can study their behavior, we can study their signaling, how they respond to neurotransmitters, how they mature, how they talk to each other, and how they look, simply how their morphology is. All that can be done with cells that are just coming from either your skin or your blood. And finally, and that will be really the most important model to study, are those mice that are 100% copying the mutation, the disease mutations. So again, you will have one copy that's good, one allele that's non-mutated, and one mutated allele, the patient-derived mutations, not just any, but we'll start with the most common and work our way, depending on how much money there is, and make these mice, and we'll describe them phenotypically, and we'll see, during development, if there are changes, what are their behavior, and also importantly, maybe for you very important, we will see whether the traits are actually rescuable. So by the time we get there in a few years, you want to know whether your children that are 5 to 10 to 15 years, whether those traits can be reversible, or they have been so much ingrained through development that they are non-reversible. It will probably, I'm not a medical person, Dr. Chung will know, but I could imagine that some of the phenotypes of the traits are amenable to therapy, and some might not, because they are too far ingrained down in the development. So that comes to the question, and I'm again not the best person to answer that, but can CK2 activity or the lack of it be compensated? So from a biochemical standpoint, that's a rare task that has not very much been done for kinases, because normally, for example, in cancer research, we want to knock down CK2 activity, we want to inhibit, and inhibitors for kinases are relatively easy to make, to design, and to modify. Activators for kinases are much more difficult, and for CK2 it will turn out a more difficult task, because CK2 naturally is already highly active. It's not really an enzyme that can become inactive and active. It is per se highly active. So to make that even more active will not be easy, but the good news is that those assays, the kinase assays that we use, they can be set up for a high throughput platform. And so a huge drug libraries that exist, that can be bought or that the companies have, can be tested relatively easily, and maybe there is one compound that could actually enhance the CK2 activity, which is what we're kind of looking for. So with this, that's the end of my talk. I wanted to thank in particular Julia, who was supposed to come here and present some mouse work, and she twisted her ankle and is unable to come, but I will tell you about her work. If you're interested, I have a very short talk that shows you the mouse behavior of the knockout mice that we made, that we believe might phenocopy, might remind us of OCNDS in certain ways. And yes, I want to thank, of course, Jennifer Sills, and you for being here and everything.